Okay. Now, silicon photodiodes are actually historically used, that means they are not put into use nowadays, and photomultiplier diodes are actually the choice nowadays. Now listen, how do this detecting system work? How do side scatter channel and forward scatter channel actually works? Now, have you ever heard of the term something called photoelectric effect? Have you ever heard of the term photoelectric effect? Now, this was actually the topic due to which Albert Einstein got Nobel Prize. Okay, but nowadays we know that his theories are not applicable. Maybe in India some classical physicist will say, classical Einsteinian physicist will say that it is wrong, but they are not put into use. It's a vague concept right now, because there were many experiments. You can see it in any book also. In biology it is a in biology it is very more complex. Einstein, if you put Einstein's concept into biological thinking, it is very much complex. It becomes very much complex because biology actually, whenever we talk about biology, we should not talk about Einstein's concept. We should always talk about Niels Bohr's concept. Okay, we have already discussed quantum physics with respect to biology in great details in other videos. You should see that. We already discussed quantum mechanics with respect to biology in great details. Okay, now listen, especially photosynthesis chapter. Now listen, what do you mean by the term photoelectric effect? Now listen, there are some metals, now listen, there are some metals. Suppose this is a metal surface, and this metal, suppose if light falls on it, okay, if light falls on it, they will eject out electron, right? They will eject out electrons. This is called photoelectric effect. Now listen, whenever light photon comes and it will eject out electron, that's why it is photoelectron. But now listen, why it is called electric? Because there is a circuit. Now listen, to understand this, just for a moment, you should understand. Suppose this is a light, this is light coming, and these are suppose electrons. Okay, now listen, you should see the circuit, that is, it is plus end of the battery. It is the plus end of the battery, right? It is the plus end of the battery and this minus end of the battery. The battery is in series. Now listen, as this is the plus end of the battery, that means this portion is actually plus and this is actually minus. Now listen, this is actually a photoactive metal, and when sun rays are falling on it or any light is falling on it, it is ejecting what? Electrons, right? It is ejecting electrons. We have discussed in great details about this in photosystem. Okay, now listen, as it is a positively charged end, that means electrons are running towards this side, right? Electrons are running towards this side, right? So when electrons are running towards this side, that means electrons are coming here and they are attracted toward this positive pole, right? Now listen, now you should understand that when electron is coming up to this surface and electron is going from here to here, that means from negative pole the electron is moving towards positive pole. That means in the opposite direction we have already got a current, right? That's why it is called photoelectric effect, right? Why I am saying this? Am I stupid? No, absolutely no. Now listen with all your attention possible. You now know what is a photoelectric effect. Right now I will be drawing the whole structure of flow cytometers, and I will make you understand why we need this concept. Listen. This was the fluidic system, right? Fluidic, generate a column of cell, single cell at a time by hydrodynamic focusing. Very good. One cell at a time. This is the laser system. It is emitting light. Cells are coming. Cells are coming. Okay. And here we have got is detection system. This is what? Forward scatter or side scatter channel? It's obvious this is forward scatter channel, right? And this is side scatter channel. Okay. Now listen with all your attention. We have up to this discussed this whole thing. Right now we will have to enter the world of electronics. Now listen, there comes the concept. Now listen with all your attention. When cells are falling, now this laser interacts with the cell, and suppose there is okay. Now both side scatter channel and forward scatter channel are now being activated. Now what are these side scatter channel? They are actually photodiodes. They are actually photodiodes. They are actually photodiodes. Now listen. That means here, from here, through a wire, electrons are coming. I am giving you the simplest explanation. We will be discussing in great detail about this in the next class. Electrons are coming. Okay, like this, electrons are coming. Now this wire actually is connected to a computer and a system. Now listen with all your attention. Now this wire is actually connected to computer. Okay. Now you should understand this basic concept right now. Listen. When these electrons are coming, now listen. This current is actually detected by this computer because of some circuit. Now listen. What you are getting is, you are getting a current, right? You are getting a current. That means you are getting a sinusoidal wave. You are getting a sinusoidal wave, right? Now whenever you will see the result of your flow cytometer experiment, you will get something like this. We'll talk about this today in great details. You will get something like this, like these, right? What are these? These are some graphs, right? That means what this computer is doing is, it is converting this sinusoidal wave into a computer graph. Graph is what? Graph is what? A digital thing. That means this sinusoidal wave has been converted to a digital signal, right? To a digital signal. This sinusoidal wave has been converted to a digital signal, and this is done by ADC. This is done by ADC. Okay. The full form of this is ADC. DC means digital converters, and A stands for analog to digital converter. ADC stands for analog to digital converter. Okay, this is called analog to digital converter. Now sinusoidal wave has been converted to digital signal, and this digital signal you will be shown in a computer. Now listen, computer has 4 options. listen with all your attention. Computer will provide you the result of your flow cytometry experiment in one of the four methods. That is, it can represent your data in a histogram pattern, in a scatter plot pattern. Okay. Now listen, there are many other patterns, two other patterns, but listen with all your attention. Today we will only discuss scatter plot, not histogram one. Okay, this is the most important thing you need to understand. We'll talk today about only scatter plot. Okay, so this is how actually your detection system actually detecting the whole thing, and it is being converted into a digital signal. Now listen, this digital signal actually is converted into a computer program by the help of a software by the name of CellQuest. CellQuest. Okay, there are many programs, many softwares which actually convert this digital signal. Let me remove it. There are many so-called softwares which convert this digital signal which is coming from sinusoidal signal. That means sinusoidal wave is converted into digital signal by the help of ADC, analog to digital converter, which is a very well-to-do name in electronics. And digital signal is now converted into computer software. Is now converted into plots, histogram by the help of a software by the name of CellQuest. Okay, now listen, CellQuest is one of the example of these softwares which actually convert these digital signals into plots and scatter plots and histogram, but there are many other softwares. Okay, so this was actually your whole idea of flow cytometer, and now we will be discussing the applications of flow cytometer, which is called flow cytometry. Okay, let's take a break, and after that we will continue with it. Thank you.